Hi everyone, good morning. Today we are going to talk about the possibility of linking the novel of Frankenstein to modern science. And we are going to discuss the topic with Professor Plazzi.
Hi everyone,
And
good morning.
okay, so can we resuscitate corpses as Dr. Frankenstein did? Is it possible?
Well, unfortunately, this is not possible actually. Obviously, it was not clear in the 19th century, but now we know that there is an aging process also for cells. During their life, the cells accumulate damages under different forms. I mean, reactive species of oxygen, DNA damages, RNA editing damages, damages to organelles. And so there is a moment when a cell becomes old. It is grew old. And so it is very difficult to reprogram it. Actually, there is an example among animals. There is a jellyfish which is considered to be mortal because in some way um, it is able to revert in its life cycle and become a polyp again. And there is a lot of research today to understand how is it possible that this jellyfish revert in the life cycle. But to our knowledge, at present date, it is very difficult to imagine that it is possible to make cells young again.
What can we resuscitate then? Maybe a part of our body or an organ, for example.
Yes, this is actually possible. Uh, to understand that, before we have to understand how the embryonic development works. Uh, we all, once upon a time were just one cell, okay, the zygote. And then there was a lot of cell division, so the single cell became 2, and then 4, and then 8, and 16, and 32, and so on. But initially, so we had a sphere of cells. But there is a moment during the embryonic development where these cells start to move, change the shape. And so the first cell of the embryo have the potential to become every possible organ in the adult stage. But very soon, actually, the cells start to start to define their fate. So immediately we assist to three groups of cells. There is a first layer which is called endoderm and will become the gut, for example, and the lungs. And those cells will become only that organs, only the gut, only the lungs, and so on. There is a second layer above the first, which will result in muscles, for example, in gonads and in other organs. So they have that fate. And finally, there is a third layer, which is the surface of the embryo that will give, for example, the skin, that will give, for example, the nervous system and the brain. And this is called ectoderm. So we start from cells that are totipotent, that can become old, and slowly they are programmed. So we have initially these first three groups, and then within the groups, some cells will define further their fate, and they will become the lungs, they will become the gut, they will become the kidney, they will become the heart, and so on. And so, as the embryonic development goes by, the more advanced is the development, the more specific is the fate of the cell. So the idea is to take the cell in the right moment when it still has the possibility to become something different and force the cell to become the organ we need.
So, do you think that we can use these cells to build an organ or even a human being?
Yes, obviously, it is very difficult to think to create a human being, but those cells are called stem cells. And as I said, they have different potential. It depends on the moment when they were taken. Okay, if I take embryonic stem cells from the sphere, for example, they have the potential to become everything. But obviously, I have to ask myself if this is possible. Okay, but is it right? Obviously, there are a lot of questions that are associated to this issue. Or I can take adult stem cells that are retained in the tissues, but they have a limited potential because they have only the potential connected to the tissue they are within. So they can only be reprogrammed to become muscles, for example, or something. Uh, Currently, yes, is it possible to cultivate those cells and obtain living tissues of the type which is requested. Obviously, it is difficult to have embryonic stem cells because, as I said before, there are a lot of issues, a lot of concerns, and we need um, a lot of a lot of thinking before doing that. To obtain obtaining adult stem cells is easier and is quite possible. Actually, we have the technology to do that and to have cell cultures in the lab.
Okay, so is this technology already feasible? Is it available already, or do you think it will be in the future?
Well, actually, yes, this technology is available. Cells and tissues can be cultivated in the labs throughout the world, and this is done normally on a daily basis, so this is not strange. It is possible to take stem cells, we are doing that, and we can use those stem cells in some cases, in many cases, to help healing, for example, of the skin when the skin is damaged by many possible things that unfortunately can happen, or to restore, to repair some organs, like, for example, the liver. And well, in recent years, there were a lot of improvements to this technology. Obviously, it is not possible to think to create ex novo a human being, a living human being. This is not possible. And maybe, I don't know, this is not right. I don't know. Because we can, as I said, especially cultivate cells of that tissue, of that type. It is very difficult to take a totipotent cells and tell it become that or that other thing. Okay. Uh, there are many concerns that are being raised. For example, we can cultivate neurons, nervous cells, there is a lot of research about that. For example, to in order to aiming to heal the Alzheimer's disease, for example. But some people is start asking, but if I cultivate nervous cells in a petri dish, when will those cells start to feel something? Maybe will they start to think? And this is a enormous question, a huge concern, and that should not be discarded too easily. Another application, which is maybe more easy, more smart, there is the possibility now to cultivate meat. Uh, the human population is very big. We are billions and billions of people, and all of these people would like to eat meat. We consume a lot of meat, and there is no space to have all this cattle. But now we can cultivate tissues from the cattle, from the cow, for example, and so we can cultivate meat in the labs. Maybe will not be exactly the same flavor, maybe yes, maybe not. But however, this would be a really cost effective and in some way really sustainable way to obtain meat for the whole human population. So also this application should not be discarded too easy, for example.
Okay, wow, it sounds incredible. It sounds very cool, but a bit complex, a bit tricky.
Oh, actually, it's not so tricky. Obviously, there is a lot of biology to understand here. Maybe you can start from our organs. So, for example, let's have a look. Let's have a look and try to understand which is the embryonic development of these organs. Now, you have here a list of 7 organs, 7 parts of the human body. So choose one, pick one, and have a look. Try to understand which is the embryonic origin of the heart, for example, or of the liver. When does it appear in the human embryo? When does it start working? When is it complete? Is it complete at the moment of the delivery, or it will be completed after? I don't know. So you can start from here, maybe. Good work.
Okay, thank you very much
You're welcome.
for coming and for the detailed analysis. Bye bye.
Bye bye. Goodbye.